So what's in this talk that, yes, unfortunately you're going to hear a lot of repetition, but hopefully there's going to, at the end I'll test you because there's probably one little tiny bit of new information and we'll see if anybody picks it out. But the idea is getting genes into plants and then seeing are they weedier. And this is an Australian bridal creeper. Can we convert that canola into bridal? Now, first of all, I want to ask a question. Is that at the end of this session, when people go out of the room, what will they do? Will they actually head for somewhere, you know, to the pub or to home? Or will they just vaguely wander about, hoping to come across something of value? Yeah. So I think that most people will, you know, have a destination of where they want to go. And so it's always handy to know what that is up front. And so following on from Alan's lead, I like to open up with my conclusions. And so therefore, there's only one problem. I have to reveal them. Could I have somebody to... Sure. Is that all right? Sorry, to put up the first slide. And the first problem is that there's actually three possible conclusions. Yeah, can unintended effects lead to increased weediness? Conclusion one is yes, two is possibly, and three is unlikely. So how do we get to those three conclusions? So I'm hoping in less than 20 minutes to do that. But so perhaps we'll go, oh, that's a duck and that's a chicken. So just for non-biological people. So if we go to the next slide, the conclusion one, yes. Well, it's pretty easy to get there. Is that the GM crop produces unintended effects. Yeah, we know that. And in the unintended effects, there'll be some unknowns. So that's fine. But if it's unknown unknowns, that'll produce aliens. Could I have the next slide? There should be, oh no, previous, sorry. There's something missing from that. Is it, and as everybody knows, is that aliens are invasive and they do bad things. And so therefore, we know that the unintended effects will do, you know, make bad things. But I would question, you know, whether they really are invasive and do bad things. And so next slide, conclusion two, possibly. And this was covered nicely in the previous set of talks. Namely, you can seek out every molecular phenotypic change and determine if one of these changes gives rise to weediness or invasiveness. And I would agree with those previous speakers, is that this is generally the way that risk assessments are done. In that, based on the substantial equivalence, we're looking for any sort of change. And then we're hoping to see if that's significant. And the issue is that we do have some examples of a small molecular change having a big effect. Is that everybody's aware of the teosinte story, you know, that teosinte is a precursor of maize. And it turns out that if you change the transcription level by 50% of one gene, you can revert back from the maize to the teosinte heavily branched state. So we know small changes. So the only problem with this approach is that we don't really know where we're heading. And the other thing is that the number of potential changes, and I've actually contributed to the literature, the number of unintended changes is actually infinite. Which is a bit difficult because my daughter's explained to me that there's actually three types of infinity, you know, the countable, the non-countable, and a third one that I couldn't understand. And so I'm not sure which one of those infinities it is, but it's one of those. And so therefore there's a lot to go through. And so therefore, it's hard to work out where we're heading. And so therefore, we're trying to take a different tack back home in Australia. And so that's the third conclusion that we're trying to head towards. Next slide, thanks. Is it trying, as I was saying at the start, rather than just going out of this room and wandering about and hoping that you come across something worthwhile or significant, is it know where you're heading. So therefore, look at all of the weediness, invasiveness traits and see if there's a significant change in those particular ones. And so that we use the Australian National Post-Border Weed Risk Management Scheme, because in Australia we've got over 1,200 major environmental and agricultural weeds. And so we have a lot of knowledge and experience of what they look like. And so therefore, if we could have the next slide. So that the value of this approach is that it doesn't matter what definition you have of unintended effect that you use. It doesn't require identification of any of your unintended effects. It provides you a decision tool to determine the relevance of any change. And so therefore, it often gets around a lot of the problems of, trying to look at every unintended change. But it requires three important assumptions. The first one we've discussed before, a GM plant is a plant. Now, outside of this room, I'd have little chance of convincing people, but even in here, I would argue that no regulator treats a GM plant as a plant. And in fact, sorry Phil, but even in Canada, I would argue that the risk assessment of a GM plant is not the same as a risk assessment of any other ordinary plant. And so this is a tough concept to try and get across, but let's assume it for the moment. The other thing is the assumption that we actually are able to identify weediness and invasiveness trait. And so I would say that can be done, and I'll go through what sort of thing that you might look at. And then does the weediness, invasiveness traits encompass all undesirable, unintended effects? And that's another assumption. And so go through some of these things is the next slide, is that what I'm going to try and suggest to you is that, because see that as a regulator, and I have to apologise, is that for, I don't have that keen scientific mind, is that I need something pretty blatant to look at. And so therefore, I would argue that if an unintended effect gives me thorns on my GM maize, then as a regulator, I'd say, hold on, there's something here that I might want to look at this a bit closer. Now, if I was really scientifically clever, David Quist type person, or someone like that, I could give you a great argument of why my trichomes that are now 0.1 of a millimetre longer are going to make me the most disastrous weeds that have ever existed. But as a regulator, I'm not very good at that, and so I need things like that. If my GM maize suddenly goes rambling, I would say that this is something I would look at. Or if its roots start extending out and breaking through walls, again. Or suddenly if my GM maize is toxic and kills Socrates again, then these are things that I can look at and say, whoa, I've got a handle. So that's what we're trying to apply. And so that, next slide. But I suppose I should just come back to what do I mean by a weed? Because there's been a lot of had some nice discussions with Andrew over this, but as to what is a weed, and there's basically a few different possibilities. Is it a plant that spreads and persists, that likes to get out there and exist? But then also that it may do bad things. And so I'm saying is it's, for a lot of regulators, is it a weed that just exists, that is invasive, that they call that a weed. In Australia, if it does bad things, that's called a weed. But you can also call a weed that has both of these characteristics, and that's what I will be looking at, and that's what the Australian Post-Border Weed Risk Assessment Scheme does. So that's how I will be using weed. And so, next slide. And so this is what the weed scientists have told us that bad plants, characteristics have. Is it a key correlation? Is the establishment. So that a plant that can establish its seedlings, survive and flourish in amongst dense vegetation is a better invader than a plant whose seed needs bare ground. Similarly, reproduction, time to seed set. A plant that comes to seeds much earlier, within a year, that's correlated with greater invasiveness than if it takes several years. Again, high seed production, more invasiveness. If it has vegetative propagules, more invasive. And the same thing is long distance dispersal. If it's more than 100 metres dispersal by wind, water, birds, other animals, and that happens often, that contributes to invasiveness. And then also human-assisted meds. So therefore, canola is one of the most invasive plants in Canada that, you must be really worried about how invasive that is. But that, humans also play a part in the whole scheme of things. And so it's either deliberate or accidental or through machinery, but also the environment can affect the spread of persistence. Is the climate and soil suitable so that affects potential distribution? So next slide is then aspects about what are the bad things that plants can do. And that generally, whenever I'm involved in GM plant discussions, is the top two get discussed. And then you ask people, what are some other bad things that plants can do? And it starts to get vague and waffly. But in Australia, there's a whole list of things that we associate as bad things with plants. That this is our values that we say are being affected. So that it reduces the establishment of desired vegetation, yield or amount, reducing the quality of products or services. Say like that, it can contaminate seed from one crop into the next rotation crop, or that the milk production is affected adversely. Or it can be, say, like the conservation status of an area goes down. That's considered a bad thing. Plants that affect the physical movement of people, animals, and water can be bad. And then there's a whole range of other things, providing food or shelter for pests or pathogens, adverse fire regimes, adversely affecting nutrient levels, salinity, stability, soil stability, water table. These are all the things that in Australia we've found and said are bad things. Now I've just given a couple of examples of, say, like the water hyacinth. Very pretty little flower, but that plant can just form a
5 minutes.
okay, so nearly there. A huge aquatic mat that stops the movement of people and boats, stops people from fishing. It allows diseases, it allows the flourish of mosquitoes and other things within those areas. The water evaporation levels go up, that the water is deoxygenated. It can end up during flood times of huge mats going downstream, breaking down bridges, fences, landing on fields, decomposing and messing up your land. And in some cases, it's clogged up systems, so that hydroelectricity systems, so that electricity to whole towns have been cut off because of this plant. This one, Chromalina, known as forest killer, which kind of gives away part of of what it does. Namely, it can grow as almost tree-like, but it can also go into rambling growth and totally smother the environment so that nothing else grows. And that it also turns out that it's a great fuel and generates fantastic fires, and so that whole villages in Africa have been burnt down. So, can I just ask one last little question? Is anybody here worried about GM plants becoming superweeds? If I were you, I would not only be scared but panicked. Because if GM maize is going to turn out worse than these ordinary everyday weeds, if they're going to be much worse than that, of closing down power to whole city, I think that's something you should be worried about, if that's a superweed. So, but anyway, so this, and I would ask you, are there any things that a GMO that could do bad, a GM plant could do bad, that is not on that list that we have experience from the weed perspective? And so that what I argue is that all of these questions, and on the previous question, is that we can work through those with our GM plant and then determine if any of those are worse in our GM plant than the comparator. And if they are, then we can put that into a risk hypothesis. So next slide, I'm just about there. Can these weed traits unmask relevant unintended effects? With past weed risk assessments, I'd say no. And so next slide. The reason is that this is how we used to do it. Is that we looked at a whole lot of we looked at one of Phil's 5 pillars. Could a plant be a weed or not? And so that with the different species, is that we looked at different characteristics, and these were different from our biology document. We were all over the place. So how do we know we captured unintended effects? And also, we didn't really have a clue of what made a weed. That was part of the problem. So next slide. And so therefore, look at those common, well understood, well known, well articulated weed traits and see if your GMO is worse. And so, second last slide, but can you tell, next slide, thanks, which ones are significant and which ones we should go for? And so just to give an example, say like canola produces about 37,000 seed per square metre per year. Now, with your GM canola, if that produces 37,500 seed, is that change significant? Well, the weeds people tell us that if a plant produces more than 1,000 seed per square metre, that's already in the high range, already making them much more prone to be invasive. And so therefore, what I would argue is that is not a significant change, because it's already producing way beyond 1,000 seed per square metre. So the other thing is that should I even bother counting the number of seed from intended or unintended effects in my GM canola when any level is still going to be in the high range from a weediness perspective? And so I think I will leave it there next. So, and just, yes, why then do we arrive at the conclusion that it's unlikely? Well, all the answers have been given before, namely the evidence from conventional breeding. Has that led to greater weediness? No. You know, what the nature of existing crops is that maize scores down here on the weed risk assessment stem. To make that a wheat, shivers, you're going to have to work really, really hard. And the specificity of the weed traits. Only very few of the changes are relevant from a weediness perspective. So last slide. I'll leave it there. Thank you. So.